Hello all, we are Team Double Diamond, and we are proud to present Project Bloom. One hallmark challenge standing in the way of mankind's colonization of the moon is that of managing food resources. While theoretically possible to bring large amounts of food on these missions, the economical costs alone make such options difficult if not infeasible. As such, we arrive at our mission statement: to investigate the ability of bacteria to grow in situ resource utilization, ISRU extracted lunar soil and assess its potential for supporting sustainable agriculture on the moon. Phosphorus is often described as the bottleneck of life, as organic phosphorus forms the backbone of DNA and is needed in large quantities to sustain agricultural productivity. However, the scarcity of phosphorus, combined with the fact that it is a non-renewable resource that is currently only mined and extracted in a handful of countries, poses not only a significant sustainability challenge for global agriculture. Phosphorus fertilizers also come with the added dimension of overuse, causing environmental degradation. On the extraterrestrial front, ISRU, in the form of extraterrestrial agriculture, is seen as the critical step that needs to be taken in order to sustain long-term human populations outside of Earth. However, this is handicapped by the lack of bioavailable nutrients such as nitrogen, phosphorus, and potassium in extraterrestrial regolith. As a result, plant life suffers in the regolith of extraterrestrial origin, posing an obstacle to extraterrestrial agriculture. One potential way to address these dual challenges is to use endolithic bacteria that can solubilize phosphorus, also known as PSBs. These bacteria convert insoluble phosphorus locked in rock into soluble and bioavailable forms that can be absorbed by plants, thereby sustaining agriculture both terrestrial and extraterrestrially. Hence, the mission of Bloom is to investigate the feasibility of using phosphorus-solubilizing bacteria to terraform the moon to support plant life, as well as to improve the efficiency and sustainability of terrestrial agriculture. To conduct our experiment, we designed an autonomous bacterial lab that can piggyback off of the Artemis 3 mission, allowing us to capitalize on multiple key benefits of the program, including logistical support and infrastructure, access to the South Polar region, and favorable environmental conditions. Although NASA has yet to confirm a landing site for the Artemis 3 mission to date, we narrowed down Nobile Rim 2, DM2, as the optimal landing site based on a multi-criteria decision-making technique that leverages lunar geographic information systems. This site offers the best ratio of explorable PSR and solar illumination, allows extended direct communication with Earth for 1/3 of the time, and is among the locations with the highest visible total lunar surface. The proximity to water ice deposits is also a key factor, with DM2 positioned nearby PSR regions for potential exploration and to support our mission. Specific to the moon, there are several challenges that any conceptual plan of terraforming for agriculture must address. As mentioned earlier, most major nutrients in lunar regolith are insoluble and locked away in forms that plants cannot easily access. These cover not only phosphorus but other critical macronutrients like potassium, nitrogen, and calcium. In some cases, lunar regolith also can contain toxic chemicals that stunt plant growth or outright kill the plants. While a significant obstacle, the dispersed nature of these chemicals relegates this factor to a secondary concern for our experiment. Other factors inherent to outer space in general are microgravity and radiation. Studies have indicated that these factors have the potential to negatively impact life in space across species. Our bacteria-based approach enables us to leverage otherwise detrimental environmental conditions, such as microgravity and lunar radiation. Microgravity impacts biological systems in ways that cannot be simulated on Earth, while lunar radiation poses significant challenges for any biological system. Some species of bacteria are even known to mutate more frequently and multiply at an accelerated rate in such space environments. While these factors have the ability to significantly impact lunar life, the factor that has the biggest research gap is arguably the lunar regolith itself. Currently, lunar regolith simulants are used to simulate the physical and chemical properties of lunar regolith for research purposes. While these are relatively easy to synthesize, the complex nature of regolith makes this approach inherently inaccurate. Actual lunar regolith, while more accurately representing the lunar conditions, is not only hard to come by but may have already been contaminated by terrestrial conditions. By studying how bacteria adapt to such hostile environments, we seek to develop resilient microbial strains that could thrive in extraterrestrial habitats while acting as a phosphorus source for lunar-based crops. Therefore, while conducting this experiment on the Moon serves to be the most accurate test of whether PSBs can serve as the foundation for lunar agriculture, the single biggest factor is the lunar regolith itself. While these factors have the ability to significantly impact lunar life, the factor that has the biggest research gap is arguably the lunar regolith itself. Currently, lunar regolith simulants are used to simulate the physical and chemical properties of lunar regolith for research purposes. While these are relatively easy to synthesize, the complex nature of regolith makes this approach inherently inaccurate. Actual lunar regolith, while more accurately representing the lunar conditions, is not only hard to come by but may have already been contaminated by terrestrial conditions. By studying how bacteria adapt to such hostile environments, we seek to develop resilient microbial strains that could thrive in extraterrestrial habitats while acting as a phosphorus source for lunar-based crops. Therefore, while conducting this experiment on the Moon serves to be the most accurate test of whether PSBs can serve as the foundation for lunar agriculture, the single biggest factor is the lunar regolith itself. The Bloom system extracts local lunar regolith near the system and cultures these bacteria in a mixture of regolith and a modified minimal salt media. The minimal salt media is modified in a way such that soluble phosphorus is the limiting factor for bacterial growth. If the bacterial cultures are then able to thrive, it indicates that they are deriving phosphorus from the lunar regolith. The bacterial species that thrive here should thus be further studied. Bloom consists of a regolith sampling robotic scoop arm, laser-induced breakdown spectroscopy system, centrifuge unit, sample activation unit, and Cartesian manipulator that interfaces with an automatic tool changer assembly that allows for the deployment of an extruder and centrifuge gripper tool head and a centrifuge tube reloader. The laser-induced breakdown spectroscopy system uses a focused high-power laser to analyze the phosphorus content of the immediate surrounding regolith prior to sampling. Although this phosphorus is likely in non-bioavailable forms, it ensures that the regolith samples have a baseline amount that can be extracted by the bacteria. Next, our regolith sampling robotic scoop arm will excavate a small amount of regolith directly from the lunar surface. A sieve ensures that only fine regolith is retained, allowing for substrate uniformity. The arm then deposits the sampled regolith into the regolith inlet system. Dual airlocks ensure that the interior of our experimental setup remains pressurized and secure when regolith enters the system. Water is then added to the received regolith and mixed thoroughly with counter-rotating impellers, forming a slurry. The centrifuge tube reloader holds the centrifuge tubes in a dispenser format. Each tube consists of pre-mixed packages of bacteria and nutrient broth in screw-capped centrifuge tubes. These tubes are stored at -80 degrees Celsius in reloader assemblies that work in tandem with our automatic tool changer subsystem. The centrifuge unit is attached to the sample activation unit. The centrifuge tube unit has the capacity to hold 8 centrifuge tubes. The sample activation unit has a vortex mixer, thermistor pads for heating, pressure sensors for vortex mixer activation, and an infrared sensor to measure the volume of regolith slurry added. Upon the start of the experiment, bacteria nutrient samples are retrieved by the centrifuge tube gripper tool and placed into the sample activation unit. The gripper tool then uncaps the tube. Our 3-axis tool head then swaps out the gripper tool for the extruder tool, feeding a standardized portion of regolith slurry into the tube. The sample activation unit then warms the sample to 25 to 30 degrees Celsius. Subsequently, the 3-axis tool head swaps the extruder tool for the gripper tool. The gripper tool then replaces the threaded screw cap on the centrifuge tube and presses the centrifuge tube into the sample activation unit. This activates the inbuilt vortex mixer in the sample activation unit to mix the regolith and bacterial culture. The gripper tool then transfers the centrifuge tube back into the centrifuge. This process is repeated for the 7 species of bacteria of interest and the control. Once all species and control setups are placed within the centrifuge, incubation occurs over the course of 7 days, after which All 8 tubes containing bacteria and the control are centrifuged at 3,000 Gs. An OD 600 reading is then taken for each of these tubes, recording the turbidity of the fluid contents post-separation. A higher turbidity reading would be indicative of a greater bacteria population, insinuating that a greater amount of phosphorus was extracted into bioavailable forms. After readings are taken, each tube is disposed of and sterilized when placed into the disposal enclosure by the gripper tool. After one cultivation cycle, the gripper tool removes the centrifuge tube and puts the spent centrifuge tube in a disposal area for destruction. These experimental results will be sent back to Earth via the Lunar Gateway through the use of our communications subsystem, including an onboard transmitter and receiver capable of interfacing with nearby relay nodes. This setup allows for both direct line-of-sight communication when the gateway is visible and indirect communication through the relay network when direct visibility is obstructed. Our power subsystem boasts a solar array system to charge both primary and secondary batteries during periods of sunlight. These reserves can then be tapped to sustain the system during lunar nights. During the extended periods of darkness, the system will enter a low-power mode to conserve energy while still maintaining essential operations. While in operation, our environmental control subsystem creates a controlled environment for conducting microbial and regolith experiments, ensuring that all life support variables are monitored and maintained automatically. Being the first lunar regolith experiment investigating phosphorus solubilization, our experiment serves as a stepping stone for agriculture-oriented microbiome engineering on the lunar surface. As climate change worsens, traditional agricultural land is losing fertility due to salinity, erosion, and nutrient depletion, threatening global food security. Our experiment uses lunar regolith as a model for degraded soils, simulating conditions where traditional farming struggles. By studying bacterial interactions with nutrient-poor substrates, we aim to develop biofertilization techniques that improve soil fertility and reduce dependence on chemical fertilizers. These innovations could help restore unproductive lands, enabling crops to grow better in harsh conditions over the next decade. This would support food security, especially in regions most affected by climate change. Additionally, biofertilization solutions from space-based research offer sustainable, eco-friendly alternatives, driving the shift toward greener agricultural practices while reducing the need for harmful chemicals. To conclude, this experiment is designed to investigate the potential of bacteria to extract phosphorus and other essential nutrients from lunar regolith, aiming to support sustainable human habitation on the Moon and provide solutions for improving agricultural practices on Earth. The scientific and technological insights gained from this experiment hold the potential to advance both space exploration and terrestrial applications. It not only seeks to address fundamental questions about the feasibility of using lunar resources to support human life, but also aims to deliver practical benefits for agriculture on Earth. By supporting the already saturated fields of lunar-based agriculture through the uncharted waters of regolith-bound bacteria research, we hope to someday see our efforts bloom on the Moon.